Yeah, so thank you everyone for joining today. I'm Ellen, and as mentioned, I'll be discussing the Lichen Cell Atlas that we've been working to develop. But before getting into what we can actually do with the Lichen Cell Atlas, how we've been developing it, let's take one step back and talk about what a lichen actually is. So many, if not all of us, coexist with lichens in our everyday lives, either on the pavements we walk on or the trees we walk underneath as we go about getting to work and going about personal life activities. And while these organisms may look and act like a single entity, in reality, they're actually a symbiotic association formed between the interaction between a fungi, the microbiont, and an algal partner, the photobiont. In lichens, when we refer to them by name, we are naming them after the fungal component. And we know that there is an incredible amount of diversity within the lichenized fungi, with currently estimated to be over 20,000 known species of lichenized fungi. And these associations come in a wide variety of shapes, sizes, and colors distributed around the globe. On the other hand, for our photobionts, the diversity remains less well characterized, but we do have a few ideas of various members that commonly occur, including representatives from the green algae Chlorophyta, specifically with genre from Triploxia and Trentipolia, as well as the photosynthetic bacteria, cyanobacteria, common representatives from the genus Nostoc. Despite their global prevalence and their ecological importance, though, the function and evolution of lichens still remains relatively poorly understood, and this has largely been perpetuated by the challenges associated with studying them. They have very slow growth rates, and we still face many challenges with actually being able to culture these in a laboratory setting, which has severely limited the path forward for how we can actually explore them. However, with more recent advances in meta-omic approaches, we've seen this big boom in the amount of lichen research and lichen data coming out. This may include long-read metagenomics, where we're able to characterize the cyanobacterial diversity across a series of different lichens, and we begin to notice co-occurrence patterns of multiple species of Nostoc occurring in one single lichen, while other researchers may also use metatranscriptomics to begin to characterize gene expression patterns found in these lichenized fungi. However, in both of these approaches, we're characterizing the picture questions about who's there and what they're doing. And this is great for characterizing them as this self-contained microecosystem, but it fails to capture the cellular diversity represented in the internal structure of the lichen, where these fungal and algal partners are organized in stratified layers, and raising the question of what functions do these different layers actually hold. As we begin to see single-cell transcriptomics being able to be applied in more diverse settings outside of your typical mammalian and model systems, this raises the question of how we can begin to use this to explore even more biodiverse contexts. We see these approaches being applied commonly now in plants to characterize different types of cell types across the developmental cycle. And more relevant to us, we've also seen this applied in the metazoan partner of corals, another photosymbiotic association, raising the question of how can we harness this technique to further explore and dig into this, the lichen symbiotic relationship. This led to the development of the project, where we had this ultimate goal of developing the first lichen single-cell atlas. Using Xanthoria paratina, or commonly known as the common orange lichen, for those of us in the UK, you've likely come across this as these bright kind of orangey things growing on rocks and trees. We use this for nuclei isolation and then single nuclei transcriptomic library preparation and sequencing. From there, we're able to analyze those reads to better characterize the different types of cell states and functions found in those individual symbionts. While the lab work represents a big portion of this project, for today, I will just be focusing on kind of the downstream data analysis side and what this means for the biological interpretation. So the fact that I'm standing here today does mean that we did get nuclei out. We do have libraries, but then the big question is, how do we actually go about analyzing this? You start with your single nuclei transcriptomic reads, And then the first step is that you will align this back to a reference genome to figure out which genes are expressed, and more specific, are expressed in which cell, or in our case, which nuclei. From there, we can generate these very large count matrices representative with the different columns representing each individual cell detected, and the different rows representing all of the different genes in your genomes. And then you do downstream analysis and you generate your very nice, colorful UMaps that we love to show at conferences like this one. So while on paper, this sounds relatively simple, you take your reads, you map them, you generate a matrix, you make a UMAP. In reality, working with the lichen data, we've been presented with a few additional challenges. One of these being a topic similar to what Paul touched on earlier, is that these are non-model organisms and we don't have reference genomes available. However, in our case, we've been able to utilize metagenomic assembled genomes instead to be able to map our reads back to. However, again, tying back into this fact that these aren't reference genomes, the quality of genome annotation can significantly impact the results. If you don't have good genes annotated, if you don't have good genomic features, how will you actually know what is being expressed? And this is kind of one of the nice outcomes of these single nuclei transcriptomic sequencing data sets. They're sequenced from the 3' end of the sequence, meaning that we can actually use these data sets concurrently to improve the genomic feature annotations of the 3' untranscribed region of the genome and be able to get out better mapping. And then finally, if these challenges weren't big enough, in our case, again, we're thinking about this in not just one species, not just one genome, but we have two that we're looking for, the fungi and the algae. This has included developing custom mapping settings for both species and doing intensive quality control to remove doublets and ambient to ensure that we're left with the highest quality data to make our biological conclusions from. So as mentioned, all of those steps are conducted twice. So now that we have this idea of how we go about analyzing the data, the big question was going to be what was actually in those libraries that we sequenced. Within metagenomic sequencing of lichens, we often have this challenge that we don't get out high quality photobiont genomes because there's just not enough coverage of them due to the fact that lichens are largely dominated by fungal biomass. However, in the case of the single nuclei libraries, we actually see the opposite occurring, where we have a very high abundance of photobionts coming out. And we also pick up some fungi as well, which has been great news because this now allows us to explore both of these partners concurrently as well. So the next step, though, was actually digging into what these individual nuclei are doing and how can we connect this back to the structure and the functions found in this symbiotic relationship. To begin with, we looked into what's going on in the fungal partner. And we've been carrying out this work in collaboration with researchers from the Sainsbury Laboratory, Nick Talbot and his postdoc, Golnera Targadzanova, where we got to the stage of being able to identify that we have 5 functionally discrete clusters, and we need to be able to figure out what this means in terms of fungal biology. And the obvious starting point here was looking at one of the more differentiated structure types found in the lichen by looking for the apothecia, which is a reproductive structure labeled here on the slide. These are the big germ tart-looking structures found on the lichen tissue. And based on previous bulk transcriptomic work they've done, they know what genes they expect to find in this tissue. So we've been able to dig into this a bit further. And by looking into this, we've been able to find some of these proteins and genes highly expressed in one cluster, cluster 4, on the left-hand side, which indicates that this is likely corresponding to that reproductive tissue that we were looking for. And further work now is going into trying to piece together what these other clusters might be contributing to in the terms of the vegetative structures. So while we have the fungi handled, we still have this other component of the lichen to consider, the photobiont. And there is really limited amounts of information of what we can actually draw upon here. These are often just simply described as photosynthetic, and they have simple morphology. People don't really look into them in high amounts of detail. So the big question was, are these photobiont cells truly only going to be serving as photosynthetic machinery, or could they actually be more functionally diverse? And if they are functionally diverse, what functions might we find in these photobionts? Similar to the fungal cells, we generated our functionally distinct clusters. In this case, we had 6. However, unlike the fungi, now we're looking into bigger picture questions about what biological processes are enriched in each of these clusters. To do this, we've been looking into differentially expressed microgenes that are cluster specific to be able to look for gene ontology or GO, functional enrichment, which is this hierarchical classification of the various types of processes. So before getting into what we've actually found within these lichen photobionts, I would just like to stress that this enrichment classification, again, isn't a binary yes or no for the presence of the function, and instead is representative of clusters of cells that show a higher level of activity for those functions. And the big starting point here, again, was looking for which cells, if any, are enriched for photosynthetic activity, because we know that that's what these photobionts should be performing. We found that photosynthetic activity was specifically enriched in only one individual cluster, with processes associated with photosystem II assembly, the photosynthesis light reaction, as well as tetrapyrrole biosynthesis and metabolism, which is corresponding to chlorophyll, our main photosynthetic pigment to be enriched in this cluster. However, the enrichment of functions didn't just stop at photosynthesis. We also see some clusters of cells being enriched for functions associated with transport. So how are these photobionts moving ions and potassium and establishing the localization of proteins? And more critical to the symbiosis, we also see a large cluster of cells being associated with cell-cell communication, and how these molecules, such as small lipids and other organic substances, may impact how these photobionts interact and communicate with other photobiont cells or their fungal partners. So just to conclude kind of some of the general information that we've had so far and what we hope to do as we move forward, as we dig into the Cell Atlas further, we've been able to explore both lichen symbionts at a higher resolution than previously accomplished with bulk transcriptomic or bulk metagenomic approaches. Using these single nuclei transcriptomic data sets, we've also been able to identify functionally diverse cell states of these symbionts to either specific structures that we know of already, or corresponding to enrichment of functional processes which may inform how these symbiotic associations are formed. But identification of these diverse cell states is not the final step in development of this data set, as we still have many questions arising now that we've been digging into this deeper. This has included how can we explore some of these more specialized symbiotic functions further. This will include additional analysis, such as using pseudo-time trajectory analysis to explore the hyphal differentiation in the fungal partner, as well as how can we use other more advanced approaches, such as spatial transcriptomics, to be able to take those microgenes that we've identified in these clusters and actually map them back onto the biological tissue to confirm where and when they are expressed. And finally, as we and other groups such as the Biodiversity Cell Atlas continue to develop these cell atlases for photosymbiosis, various types of photosymbioses, it raises the opportunity to eventually conduct integrated comparative analysis to be able to look for conserved functions found across these diverse photobionts rising from different lineages such as chlorophyta and dinoflagellates and associated with different host types. And finally, just to conclude before questions, to acknowledge all of the groups that have been supporting this work, and I'm happy to take any questions.
Thank you. So any questions? Oh, yes. I'm just going to get first, Paul. Great.
Hi, thank you. That was a really interesting presentation. I imagine working with lichen is a bit of a nightmare. It doesn't grow easily, and I don't know if you can grow under sterile conditions. And I also remember hearing that the bacteria associated with the fungi are often far more diverse than just the photobionts. And so I'm curious about the diversity of the bacteria you found and how that influences your analysis of the single cell data.
Yeah. So for the first bit, we don't we do the very scientific thing here, and we don't grow them, and we just go outside and pick them up off the ground. So yeah, those lichens are just from outside the Sanger Institute. And then for the bacteria, so today I was only presenting on the single nucleotide transcriptomics, and unfortunately, the way that these data sets are generated, first of all, the nuclei, so bacteria won't be included, and then the sequencing technology is normally poly-A tail based as well. So again, the bacteria are lagging a bit behind. I know that there are people starting to do this for bacteria. I think that would be really neat to have that additional layer in. But yeah, there's in the metagenomes and metatranscriptomes, they show up, but not in the single nuclei at this stage.
So yes, this is really cool work showing us how a lichen can work. I guess the question is, how do you know your model system is in any way typical of how lichens actually work out there in the environment? Have you just picked a weird one, which is doing something completely different to 90% of other lichens?
So we have 4 different biological lichen replicates, so found on different sticks outside of the Sanger Institute. So at least we know that some of these trends are maybe it's just the Wellcome Genome Campus, but obviously as well, looking at just Xanthoria paretina is not capturing the whole diversity that we see in lichenized fungi, because we have different morphologies, different photobionts. So this is really just starting to scratch the surface, but it's, the start.
The Wellcome Genome Campus isn't representative of the rest of the world normally. The real world. That's in there. Okay. Thank you very much.